Assalamu alaikum. My name is Hamna Shams from the department BSBT. My today's topic of presentation is spinach. Introduction to spinach. Hyperlipidemia, a condition characterized by high levels of circulating fats, is regarded as a modifiable risk factor for cardiovascular diseases and cerebrovascular diseases. The incidence of these vascular disorders among South Koreans is increasing at a rapid rate due in part to adoption of a more Western lifestyle and increased consumption of fatty foods. A high-fat diet has been shown to increase oxidative stress in a variety of tissues, a side effect that may contribute to the development of numerous degenerative diseases. In order to prevent or moderate oxidation-related diseases, it is necessary to sequester and eliminate free radicals from the body. Antioxidants may offer some resistance to oxidative stress by scavenging free radicals, inhibiting cell membrane damage, and suppressing lipid peroxidation, thus preventing the onset of chronic diseases. Materials and methods. Sample preparation. Spinach in dried powder was obtained from the Spinach Cluster Agency after being cultivated in Shinan, Iceland, Jeonnam, Korea. For preparation of the hot water extracts, 5 grams of spinach powder was put in a glass bottle with 100 ml of deionized water, extracted with an autoclave and freeze dried. The ethanol extracts were prepared as follows. 5 grams of spinach powder was extracted with 100 ml of ethyl alcohol and concentrated by evaporator. We and EE were redissolved in dimethyl sulfoxide to a concentration of 50 mg per ml, and aliquots were kept at 20 degrees Celsius until used. Measurement of total phenolic contents. Total polyphenol content was measured by the method of a POC ETAL. Briefly, WE was mixed with 2 ml of 1 normal Folin reagent and incubated at room temperature. Then 2 ml of 10% sodium carbonate were added and the mixture was incubated at room temperature. The absorbance was measured at 690 nanometer with an absorbent enzyme linked immunosorbent assay. ELISA reader. Oxygen radical absorbance capacity assay. The oxygen radical absorbance capacity assay was carried out on a flu star of optima fluorescence plate reader with fluorescent filters. Excitation wavelength 485 nanometer, emission wavelength 535 nanometer, according to the method of Park et al. The results were calculated based on the difference in the area under the fluorescence decay curves between the blank and each sample. Oricro was expressed as micromole of Trollocs equivalents. Cellular antioxidant activity assay. Hep G2 cells were seeded at a density of 5 × 10⁴ cells per ml on a 9-6 well microplates. 24 hours after seeding, the growth medium was removed and the wells were washed with phosphate buffer saline. Triplicate wells were treated for 1 hour with 100 microliter of extract dissolved in treatment medium. Wells were washed with 100 microliter of PBS and 80 micrometer of AAPH in 100 microliter of PBS plus 40 micrometer DCFH DA was applied to the cells. Following the condition of the AAPH solution, the 96 wells microplate was placed into a flu star optima fluorescence plate readed at 37.8 degrees Celsius. Emission at 535 nanometer was measured with excitation at 485 nanometer. Triplicate control and blank wells were included on each plate. Control wells contained cells treated with DCFHDA and an oxidant. AAPH blank wells contain cells treated with PBS without AAPH. Blood and hepatic lipid profiles. Plasma lipid profiles, total cholesterol, high density lipoprotein were measured using assay kits from bioclinical system and a photometric autoanalyzer. Plasma low density lipoprotein cholesterol concentrations were calculated using the formula developed by Friedwald et al. The concentration of total lipids in liver samples was determined using the method of Fock. Total cholesterol and triglyceride concentration in liver samples were analyzed with the same enzymatic kits used in the plasma analysis. Baseline conjugated dienes in LDLS. The concentration of conjugated dienes in plasma was determined according to the method of Pock et al. Briefly, plasma was added to 700 microliter of heparin citrate buffer and incubated for 10 minutes at room temperature. After centrifugation at 1000 gram for 10 minutes, the pellet was resuspended in 100 microliter of 0.1 mole sodium phosphate buffers containing 0.9% sodium chloride, which had a pH 7.4. Lipids were extracted from 100 microliter of the LDS suspension with chloroform permethanol 2:1 dried under nitrogen and redissolved in cyclohexane. A spectrophotometer UV1205 was used to determine the absorbance of the redissolved sample at 234 nanometer. Now the hepatic thiobarbituric acid reactive substance. The thiobarbituric acid reactive substance level in plasma was estimated by the method of Big and Ost briefly. 0.2 ml of plasma was mixed with 0.3 ml of distilled water and added to 1 ml of mixture containing 0.38% thiobarbituric acid, 15% trichloroacetic acid, and 0.25 normal HCl. The resulting mixture was incubated for 20 minutes in a boiling water bath prior to centrifugation at 2000 grams for 10 minutes. The absorbance of the supernatant was measured at 540 nanometer using 1133 tetramethoxypropane as standard. Lipid peroxidation was expressed as TBARS in n mole per ml plasma. Liver lipid peroxidation levels were measured by the method of Ohkawa et al. With some modification. Liver homogenates 0.4 ml in 1.15% KCl, 0.1 ml of 8.1% Astria, 0.75 ml of 20% acetic acid, and 0.75 ml of 0.8% TBA were added to a test tube. Then the sample was vortexed and heated in a 95 degrees Celsius oil bath for 1 hour. After cooling for 10 minutes, butanol pyridine 15:1 volume to volume ratio was added. The sample was mixed thoroughly and centrifuged at 3515 grams for 15 minutes. The fluorescence of the upper layer was measured at 552 nanometer. The amount of MDA present in the sample was converted to TBARS values using a TMP standard curve. A Pierce BCA protein assay kit was used to quantify the protein concentration of each liver sample. Erythrocytic catalysis. Erythrocytic hemolysates were prepared by the deletion of erythrocytes to 1:500 was distilled water. 100 microliter of erythrocytic hemolysate was dissolved in 50 mm phosphate buffer, 50 ml pH 7, and 2 ml of the mixture was added to a cuvette. The reaction was initiated by the addition of 1 ml of H2O2 30 nm at 20 degrees Celsius. The H2O2 decomposition rate was measured at 240 nanometer for 30 seconds using a spectrophotometer. Hepatic antioxidant enzymes. To determine liver superoxide dismutase activity, liver samples were homogenized with 0.1 ml of 65 mm phosphate buffer, which had the pH 7.8, and then centrifuged at 10,000 grams for 20 minutes. Copper zinc superoxide dismutase activity was measured in the resulting supernatant. The remaining pellet was dissolved in 0.1% tritone and used for the determination of manganese superoxide. dismutase activity. For the MnSOD activity assay, 4 mole of KCN solution was added to the assay mixture to inhibit copper zinc SOD for MnSOD and Cu per Zn SOD analysis. The samples were pre-incubated with 75 sodium xanthine and 10 hydroxylamine hydrochloride at 37 degrees Celsius for 10 minutes. Then 0.1 units of xanthine oxidase was added and samples were incubated at 37 degrees Celsius for an additional 20 minutes. The reaction was stopped by the addition of 1% sulfonamide and 0.02% ethylene diamine dihydrochloride. After standing at room temperature for 20 minutes, the absorbance of the final mixtures was made at 540 nanometer. 1 nitrate unit of SOD activity was defined as the amount of protein required for 50% inhibition. Catalase activity was made according to the method of Carrillo et al. Liver tissue was homogenized in sodium-potassium phosphate buffer which had the pH 7. The homogenates were centrifuged at 600 gram for 10 minutes to obtain the supertet. After centrifuging at 10,000 gram for 20 minutes, the supertet was discarded and the pellet was suspended in 1:RBC buffer. After incubating on ice for 10 minutes, the suspension was centrifuged at 10,000 gram for 20 minutes. The pellet was washed in sodium-potassium phosphate buffer which had the pH 7 and centrifugation was repeated. After washing, the pellet was resuspended in sodium-potassium phosphate buffer. For the acid, sodium-potassium phosphate buffer and sample were mixed and Curtis covet, and the reaction was started by the addition of 300 microliter of 30 H2O2 solution. The H2O2 hydrogen peroxide decomposition rate was made at 240 nanometer for 40 seconds using a spectrophotometer. Glutathione peroxidase activity was made according to the method of Bogdanska et al. Liver tissue was homogenized with 1 ml of 250 potassium phosphate buffer which had the pH 7 and then centrifuged at 10,000 gram for 20 minutes. 25 microliter of supernatant containing cytosolic fraction was incubated with 10 EDTA, 10 NaN3, 10 GSH, 2 NADPH and 1 unit glutathione reductase at room temperature for 5 minutes. The reaction was initiated by the addition of 25 microliter of 2.5 H2O2. The H2O2 decomposition rate was made at 340 nanometer for 70 seconds using a spectrophotometer. The protein concentration of each supernatant was determined by BCA protein assay. Samples containing equal amount of protein 25 microgram were used for the enzyme activity assays. DNA damage in rat leukocytes. 20 microliter of whole blood were suspended with 150 microliter of 0.7% low melting agarose and added to slides that had been pre-coated with agarose. The slides were then treated in PBS with 200 micro H2O2 for 5 minutes on ice and washed with PBS. The following steps were the same as done in the in vitro DNA damage determination by Cormet assay. Statistical analysis. All measurements were analyzed using the SPSS package for Windows. Mean values among concentration per animal groups were compared using one-way analysis of variance followed by Duncan's multiple range test. Results. Total polyphenol content, antioxidant and anti-genotoxic effects of spinach. The TPC of WNEE were 1.5 plus minus 0.0 mg GAE per gram and 0.5 plus minus 0.0 mg GAE per gram respectively. The auric values of WE and EE increased in a concentration dependent manner at the highest tested concentration 50 microgram per ml. W 7.6 plus minus 0.1 T and EE 7.2 plus minus 0.1 T showed similar ability to protect a fluorescent reporter from oxidative degeneration by AAPH. The viability of spinach extract related HAPG2 cells was 70%, the highest concentration tested. 50 microgram per ml data not shown. These extracts reduced AAPH. AAPH induced oxidative stress in HAPG2 cells. Figure 1b is basically the link at this statement with high concentration producing greater increasing in CAA. Pre-treatment of the human leukocyte with spinach extract for 30 minutes significantly reduced hydrogen peroxide induced oxidative stress, with high concentrations showing increased ability to inhibit DNA damage. This one is basically the graph showing the above statement. Now, effects of spinach on blood and hepatic lipid profiles. In the in vivo studies, no adverse reaction or clinical signs were observed in HFCD rats throughout the feeding period, as shown in table 1. You can see here. There was a marked increase in the levels of TC and LDL-C in the HFCD group compared with ND group. Plasma HDL-C decreased significantly in the HFCD group compared to the ND group. Plasma TC, HDL-C, and LDL-C levels did not differ between HFCD and HFCD plus S rats. Additionally, while hepatic total lipid, TC, and triglyceride levels were significantly higher in the HFCD rats than in the ND rats, these concentrations did not differ between the HFCD and HFCD plus S groups. Effects of spinach on antioxidant status. Liver TBARS levels were 79% higher in rats in the HFCD group versus the ND group, as shown in figure. Liver TBARS levels in rats that received the HFCD plus were not different from those of rats that received the ND. There was no difference in plasma TBARS level among treatment groups. These are the references. These are basically 10 health benefits of spinach. You can see here. Number 1, spinach is good for asthma patient, help in eye health. Spinach help in weight loss, prevent acne and help to glow skin, prevent anemia problems, helpful in brain functionality, helpful in boosting immunity, helps to make bones stronger, benefit in diabetes, and reduce the risk of cancer. Eat spinach and be healthy. These are the unexpected side effects of spinach. Number 1, may increase kidney stone. Eating too much spinach may cause kidney stones due to the high oxalate content. Number 2, may interfere with blood thinners. The high levels of vitamin K in spinach may reduce the effectiveness of blood thinners. Number 3, may hinder mineral absorption. The oxalates anti-nutrient in spinach may inhibit mineral absorption. Number 4, may aggravate symptoms of gout. Spinach contains purines, chemical compounds that may contribute to gout. So this one